Hello everyone, I welcome you to another interesting session of Mendel's bicentenary celebrations. I'm glad to introduce myself. I am Mercy Rofina, pursuing my PhD in Dr. Vinod's Careers Lab at CSIR IGIB. For today's session, we have our speaker here, Dr. Arindam Maitra, who is the officiating director of National Institute of Biomedical Genomics, NIBMG, and he's also the project coordinator for International Cancer Genome Consortium. It's a pleasure to have you here in this platform, sir. So his research focus is majorly in exploring human disease genomics, and he's here to share his thoughts and opinions about genetics and how it has influenced in shaping our national health policies and what are the path forward. So we welcome you to this session, sir. So, like I told, we know that you've been working in exploring the genomics of extensive genome analysis of various genetic disorders. So, it would be great if you could elaborate on what exactly is your research interest or what your lab is currently now focusing on.
Thank you. Thank you for inviting me for this. This is very interesting. And definitely, it's a very nice way of looking at Mendel's 200 years after Mendel. Glad to be part of it. So before I tell what my lab works or we work right now, so let me give a brief introduction about myself. I am basically a student of chemistry. And after I did graduation in chemistry, I got into biochemistry because that's what fascinated me. And subsequently, PhD onwards, I was fascinated with the way genetic variation plays a role in human health and disease. So started my PhD in all in medical sciences with HIV sequencing, HIV genome. At that time, way back, it was very challenging. And then we went on and got dabbled in the industry again with sequencing, genotyping, and understanding how variations tools can be used to harness these variations in the human genome, and subsequently got back to basic research, where entire career I have spent on human disease research, looking at understanding genetic variation and how that plays a role in human health and disease. So right now in IBMG, before I tell about what we do, let me tell you about the institute. The institute, again, is like Institute of Genomics and Integrative Biology, and IBMG is also a genomics institute. That means we do not do anything else except genomics. And we are a very focused genomics institute. We only work on genomics of human health and disease. So we are completely focused on understanding how genomic variation plays a role in human health and disease. And hence, to all of us, Mendel has a very special place in our heart, because nothing of this kind would have been possible had those Mendel's laws been not there and his experiments with the peas had not happened. Probably we would not have been here now where we are.
Yeah.
The institute was set up about 12 years ago and works primarily on 4 broad areas. We study genomics of cancer, various cancers. And we study genomics of other complex diseases. We also study genomics of infectious diseases with a particular focus on the host, although also that does not rule out looking at correlating genomic variations in the pathogen genome itself. And there's a very vibrant wing we have here, which is on statistical and computational genomics, which is essential to fully interpret and understand and analyze genomic data at the scale that in modern genomics we handle right now. Now, giving that backdrop, I tunnel down to what I do or we have been doing in NIBMG. So I am involved in 2 areas. So my whole focus is that understanding how genomic variation plays a role in human disease or human health. So what makes us keep healthy or what makes us fall ill? And there are 2 broad areas where I work. One is genomics of head and neck cancer. Here, my group is trying to understand in head and neck cancer, particularly oral squamous cell carcinoma of the gingiva buccal or OSCGB, which is the most predominant form of oral cancer in India. we are trying to understand what are the genomic, epigenomic, and transcriptomic alterations that drive this cancer, and what are the signatures of these kind of information that will help us to classify patients into understanding whether, pre-cancer, patient with a pre-cancer will develop into cancer, or whether patient with cancer who is treated will have a quicker relapse versus those that have a low chance of relapse. Of late, I have been deep diving into the tumor microenvironment of oral cancer by using single cell transcriptomic approaches. And right now we are all fascinated by in our present work of trying to unravel and illuminate the tumor microenvironment, understand what are the different gene expression programs and how do they vary between individuals and cells in this tumor microenvironment. Right. So this is one area. The other area that I work on is maternal child health, where I and my colleagues are trying to identify what are the genomic alterations, and my genomic, I also include epigenomic and transcriptomic alterations that underlie prematurity or preterm birth, which is again an important public health problem. Babies born preterm not only have a very high mortality, infant mortality, have delayed developmental milestones, but also have a higher risk of adult onset diseases. So has an enormous public health connotation. So in this, we are, as I said, working on a large pregnancy cohort, trying to understand these things. We are doing genome-wide association studies and large-scale longitudinal studies to understand these things. Of late, again, working on placenta, again, deep diving into placenta, trying to understand using single cell approaches how placental cells interact in terms of gene expression and which of these are important for prematurity. So this is what we do, and also love to do many outreach.
Right. That was really indeed very interesting, sir, to know the different fields of your focus. So since you have mentioned that you've been working on cancers, so I would like to ask, you would have really seen not just the sporadic cases, you would have seen cases running in families, you could have met with some families. Do you think there's enough amount of awareness about this term of heredity or the disease or a variant running in a family? And what do you think about the stigma that comes along with that? Like, what is the level of awareness among a general public? And what do you think is important for a student to dig deep into a genetics field? If you could share something on that note.
I will first answer what you directly asked, and that is, what about heredity? What is the role of heredity in cancer? We know that for certain heredity does play a role in cancer, but only a small proportion of cancer. Because if you go back to the Knudsen's two-hit hypothesis, so if you have a tumor suppressor gene, I mean, it takes 2 hits to knock off the gene in a particular cell, because a cell has 2 copies of the gene. What happens is, in inherited cancers, you already have one of the alleles knocked off, and that is inherited. And then by environmental exposures and so forth, you have the other. So it is a shorter route to the initiation of the cancer. I think there has been traditionally very little understanding of cancer, particularly these kind of things in society. But of late, there has been substantial progress, particularly clinicians have now are more conversant with genetics, and they are more aware of genetics, and they are started to use genetics in clinical practice. Although, I mean, in India, a lot more needs to be done, but still it's there. And in certain cancer types, for example, it has been very well, remarkably worked out, like in breast cancer. So for breast cancer screening, I mean, the routine question that the clinician asked is for the woman who was in the family, is there any aunt or mother or anybody who had breast cancer? And they can actually calculate in front of the patient and explain to them what is the risk of developing breast cancer, given the family history. So these are very good stories. So now these are quantitative understanding of cancer risk of in inheritance, and these are probably very success stories that have come up. But more importantly, so how do you look at heredity? I mean, if you look at individual generations and individuals and look at heredity, there is another kind of heredity that is between cells. So in an individual, of late, we are now more understanding that we have to, beyond taking individuals as units, we need to look at cells as units and take genetics to the cells. So 90% of the cancers that we face or encounter are sporadic in nature, which means that they do, the cancers develop because of an environmental impact on the cells and during the mutations that occur during the mitosis, which is the process that happens within individuals during the lifetime when cells give rise to progeny. And there is heredity there as well, because information is transmitted from one cell to the next within the individual, and somatic mutations that occur and result of environmental impact and so forth, they then fuel carcinogenesis or tumorigenesis and then result in a full-blown cancer. So these are various aspects of this. One is little more difficult to explain and for people to understand, but we had been taking a lot of outreach programs and so forth where we try to explain to students, even high school students and college students. College students now know a little better. The curriculums have vastly improved, but even to high school students and their parents that, look, this is how cancer happens.
Right.
So that's been of late. I think there has been a lot of progress, although a lot more needs to be done.
Yeah, so on the same note on what you have mentioned, so for this program also, we have been in talking to some undergraduate students from medical or engineering or science. So, and I've been asking why did they choose biology or why they didn't choose biology. One of the response I was getting constantly was biology is a tough subject and it involves a lot of memorization. We need to remember the names, remember the principles. So I think there's something which should be added in the education to create more interest, like you mentioned, among the college students or even in school education. They should be inclined towards a more interesting way of learning biology. Do you have anything to say about that, sir?
Yes, I think I have always loved biology, but then I've also loved physics and mathematics and chemistry. But the point is, so what I find fascinating about biology and what I find is still missing from our curriculum, right from school days, to explain to students and present how we present biology to students. Biology, essentially the most interesting part of biology, which we are now seeing because we are able to see biology at that resolution and depth today, is because of its interdisciplinary nature. So that interdisciplinary nature, that biology consists of mathematical principles, biology consists of principles of physics in action or chemistry that is happening within the cells. That part needs to be brought forward, and I think the students will find that fascinating. Exactly. That is foremost on how biology needs to come out of textbooks into these kind of explanations. And also biology makes more sense when it is shown in action, giving real examples, making simple experiments in the labs and showing what biology means. I mean, DNA, it's very difficult to teach students DNA, but you just ask them to, take a little blood or spit saliva, do a sorting out experiment and see your DNA strands. And then from there, it goes stepwise. So these are the approaches which will probably make biology more fascinating. And for senior students, like, which are in colleges levels, I think the interdisciplinary nature needs to be brought out more for biology, because biology is most exciting in its interdisciplinary mode rather than, just traditional biology per se, how it has been taught in textbooks.
True, very true, sir. I completely agree with that statement that interdisciplinary aspect should be like seated in a very initial stage when the student is studying about biology. So my next question is, since you've been in this field for so long and you're working on something which is translationally very, very relevant on a medical setting, So what do you think is needed to, or what advancement is further needed now that the sequencing costs are also coming down? What exactly will be the future need or one step forward, which will lead to a better way of shaping our health policies or as a nation, or moving towards a better way of living, better quality of life? So, what would be that,
Yeah.
according to you?
So I think there are many things that can be done, but I will probably mention two of them. One is look at health or health cohorts,
Mhm.
cohorts which are for a particular health trait or a disease trait, and do sequencing of these cohorts to understand and map and catalog all the genetic variation, and so that use that data to identify the underlying variations that result in this kind of diseases. So forming trait-based cohorts, sequencing these cohorts is one thing that I should think that should happen. And the second is, of course, our recent understanding, as I was saying, that looking at, once you look at individuals, the second dimension of deep diving into, so we are more realizing that individuals as units, we look at cells as units. Even
Yes.
within an individual, within a particular tissue or an organ of the individual, there are millions of cells, and each cell is different. For an example, I mean, you have 1,000 people singing a song in chorus. You get an average readout. You do not, I mean, you do not know what are the individual variations, which are
Right.
probably important. Three bad singers can completely make a very poor performance, whereas five good singers can probably compensate that. But how do you identify them? You cannot from the chorus. You have to listen to, you have to deconvolute that chorus into individuals' own performance and identify this. So single cell genomics. So looking at cohorts, sequencing, more sequencing, sequencing the individuals, and also looking at single cell genomics to answer some of the health traits and disease traits are two things that I should mention. This is the way forward.
Very, very one of very interesting opinions, sir. So, I was doing interviews of many other people also as a part of the series, and one thing I got to note was certain kind of illness being prevalent in some groups, in some subpopulations, or in some geographical locations. So, obviously, we get to know that there is a differing prevalence in the diseases in different parts. And if you had noticed anything like that, a separate subgroup or something where there is more prevalence of one particular disorder, and do you think the people are aware of that? Is enough outreach being given to those people? Anything on that note, if you have any opinion to share?
Yes, as you know that I mean, in IBMG and I particularly have a lot of collaborations and engagements in the northeast part of the country.
Yes.
And we have seen that there are, for certain cancer types, the northeast has a very high prevalence. So and we understand that there would be environmental exposures which are characteristic of that region. There would be, it's a little complex. So there are the environmental exposures, and as well because of consanguineous kind of population structure in some of these areas, there would be a inherent risk is also probably high. And I had been trying to work in head and neck cancer and in gastric cancer, working in the northeast in certain regions where the incidence is very high, and we are trying to sequence and do genetics to understand what are the differences here. I mean, what are the unique genetic variations which are prevalent in this population in this area, which is actually probably is different from the rest. So one example is oropharyngeal cancer, and we found in Meghalaya particularly it's very high because people eat these areca nuts, raw areca nuts, and it is directly associated. So we had been doing some genetic study, genomic studies there, and also in gastric cancer in Mizoram, where we know that there are certain risk lifestyle, food habits which are more. And then this right now, I think earlier there was not much awareness, but all this is leading to awareness. So even sometimes genetics gives a feedback. So when we do these sequencing studies, we identify mutational signatures for certain mutagens.
Right.
And that is related to maybe the areca nut, or is related to the smoked meat or fermented meat that is used by individuals more often in these areas. And that leads to the doctors are very eager, the clinicians are there very eager to understand all this. And they are very active in trying to explain to their patients and to people in those areas that, look, these are the high risk factors and that needs to stop.
Right. So it was really interesting, sir, like knowing about the dietary practices of the people and checking back there if there are any genetic markers. Also, you had stated one of the most unique points about the single cell genomics, which needs to be established in the nation, which needs to be every science which we do right now needs to be done in a very, very detailed way. That was one of the most interesting points you have stated in this session. So coming to the end of this session, I would like to ask you if you have any a kind of a motivation message or a kind of encouragement which you want to tell for young researchers who are currently working in this field. Like, what do you want to tell them as a senior investigator? Like, what message you have for students?
So one thing I would definitely encourage all of you young people who are working in this area is to appreciate and understand the interdisciplinary nature of your research. I mean, explore on that. Uh, dig deeper, and you will only be able to do that if you use such approaches, using mathematical approaches in genetics, using various other kinds, collaborating with people who are from a different field, but who can make more, who can provide a particular sense or meaning to your genomic discovery or similar discovery. So those kind of collaborations and tie-ups, these are very important, and start them from very younger age. I mean, don't depend upon your teachers or supervisors or people to do that. You yourself start developing that kind of interaction, because that will make you appreciate and make most of whatever results you are getting, whatever research you are getting. Second is, I mean, do not let your motivation fall, because whatever you are doing it's absolutely cutting edge. And many of this, if not today but tomorrow, will probably, if worked upon adequately, will lead to some betterment of condition of individuals, so and some betterment of the system. So these are the two things that should keep you motivated. And don't, I mean nothing is difficult, and nothing is difficult and nothing is easy in a sense, because yes, if you have to do something, the more impactful that work is, will be also be more challenging. So you will have more obstacles. But then again, if you use different kind of approaches, think innovatively, come out of the box, interact more with others, and try to get more sense, you should be able to overcome these challenges. And that's why I said it is easy in the end.
Right.
Thank you so much.
Thanks a lot, sir. It was really a very insightful discussion with you today. And we are very glad you accepted the invite and took time to share your thoughts and opinions about all the questions I had for you. Thanks a lot for joining us today, sir.
Thank you. Thank you so much.
Thank you, sir.
Bye-bye.